several conferences, seminars, workshops, participated, presented. Sir also presented several papers. There are 13 papers published. There are 2 books published. Sir also acted as a resource person, guest lecture, chair, co-chair, technical speaker in so many workshops and seminars. Sir also board member, Yashwantrao Chavan College of Science, Satara, India, Botany Department, Sandeep University, Nasik, India. Sir also life member of the Bombay Natural History Society, Mumbai, India, and Indian Association of Angiosperm Taxonomy, Calcutta, India, Systematic Association, London, UK. Sir also visited several nations, that is including United Kingdom, England, Royal Botanical Gardens, QN National History Museum, London, Dubai, Saudi Arabia. This is very briefly about this, sir. And you can reach out this, sir, in the Scopus. Scopus ID is also there. We will share the ID. You can see this, our scientific contribution, and in the Scopus, in the Google also. Yeah, this is with this few details. I welcome Nandikar sir for the 1 week short-term course on plant taxonomy on behalf of the organizers and the participants and Usmania University. Once again, I welcome you, sir. Thank you.
Yeah, thank you, Nageshwara sir, for this brief introduction of mine. Dr. Reddy wanted to tell something, or should I start with the session?
Unmute.
Yours mic.
Yes, you can start, sir. You can start, please. Please start, sir. Yeah.
So thank you once again, all the staff of Nizam College. Yeah, my voice is audible to you all, I believe. And thank you very much, the staff and the authorities of the Nizam College, that they have given me an opportunity. Secondly, I thank Dr. Chili Pays over here. And I also thanks our center that they are allowing me to do such kind of stuff. And life becomes so virtual. I mean to say, last 3-4 months, we all are experiencing the same. But the field of taxonomy cannot be virtual at all. As I have spent almost 13-14 years in this group, and I'm very happy to present all the things in front of you. Almost, I believe, more than 100 participants are there today. In fact, if we wanted to have a physical kind of meeting or the webinar or the workshop of organizations. So I spoke that day. I was talking with Dr. Reddy that we can accommodate as much as people we can, because it is the platform that everyone can use while sitting there at home. So I'm not taking much of your time. I'm going to present today that very close to my heart. The topic is called family Commelinaceae in India. We call it as the spider words. And as I told you, since last almost 13-14 years, I'm working in this group. And always I'm excited to present the Commelinaceae. As I told, it's very close to my heart. Yesterday only I have prepared one rakhi made up of this Mudania striated petala. So this is the tiny flower that we rediscovered from the eastern coast, from the Chennai. It was known from Sri Lanka earlier. And this is what Indo-Sri Lankan endemic, and has a unique flower with striated petals. So I'll come to the genus called Mudania. So for a basic, I will talk something about the Commelinaceae. You may know the Poaceae, you may know the Ericaceae, Gingiberales, various gingers, and other families belong to Commelinaceae. I wanted to trace over here family Commelinaceae or the Commelinaceae. The clade Commelinaceae is like a consistently evolving clade. some of the apomorphic character. For example, here it is breaking. From here it is segregating. All the families listed over here. This is following APG Angiosperm phylogeny group recent classification, where the Commelinaceae is occurring over here. And if you can make it out, it has been separated from all the Commelinaceae, remaining of the Commelinaceae, due to have the glandular microhairs. And rest all the families are like brought together because of their having specific organic chemicals. As you know that they are like separated. If you ask me as the basic question that why the poels and the arachales has been clubbed under the Commelinaceae, because all are sharing the common ancestral character, that is having the typical organic chemicals. And in other words, for example, if I take the Commelinaceae or the Commelinaceae over here, we are very acquainted with the Commelinaceae as well as the Pontederaceae. You know the many members of the Pontederaceae like Ecornia, Pontederia and all. Similarly, we also have Commelinaceae. Popularly, we call it as a spider-verse and has a plants called Tradescantia. That is, everyone might be aware with it. Commelina is there. Cyanotis is also there. So this is what entire structure of Commelinaceae. And if you ask me personally, when I joined this subject in 2007, when I joined for the MPhil, I realized there are a lot of work happened in the past during 1960s, 1970s by Dr. Sheshagiri Rolarao, Kamathi and other people. And there is nothing to do in this family, because I was having the belief that already people have worked and they found many interesting findings in their work. But unfortunately, the other parties, I found there is no entire comprehensive account of the families available at that age. Then I thought we need to work out, and that's how the work has been started to make it the family more comprehensive for India. And today we reached over around 98 species belonging to 15 genera, including ornamentals. And as I told already, Commelinaceae is called as the day flower or the spider birds. Day flower because flowers are having very short lives. They bloom for a few morning hours and they will fade up. And family total over the world, it was known of 41 genera and around 650 species. And it has a very diverse in morphology. If you see the status of the word distribution of this family Commelinaceae is divided into the sub two families like a Tradescantia and Commelini. That Commelini is more dominant in the old world. This is what, and the tribe or Tradescantia is more dominated in the new world. So if you take genera called Tradescantia, Calesia, Tinansia, all the genera are properly distributed in New World, while some are distributed in the Old World. For example, many distribution with the New World as well as the Old World. I'll come to India quickly. This is what the overall distribution of the genera in India. Peninsular India, or you can say call it as the Western Ghats, is more dominated with the tribe Commelini, and these are the genera I have listed over here, including Belosynopsis, Dictyospermum, Commelina. Whereas northeastern parts and Andaman Nicobar Islands having more number of Tradescantia members. I don't know how much are you aware with the Commelinaceae genera. Most of you are well aware with the Commelina, Cyanotis, Tradescantia and all. This is the entire enumeration of the genera. Total number of species around 98. Now it crossed almost 102. And there are 26 endemic species are there, endemic taxa we can call. And this is how where we are landing. So as I have started is the Commelinaceae, Commelinoidy, and Taxonomy, Commelinaceae. So this is what you can make it out the flower over here. It has a typical petals with the proper symmetry. If you have the section of the two equal, you will get the two equal parts. But where in Commelinaceae you will find sub-equal petals. For example, these two lateral petals, and here inconspicuous medial petal. So if you have if you have a section in the middle, you will have the bilateral symmetrical, but the entire flower or the petals are not symmetrical. So here, number of fertile stamens, you can make it out the dots over here, 1, 2, 3, 4, 5, 6. But here, number of fertile stamens or 2, 2, 3. It means that there is whirl of staminodes too. I will start with the habit. Commelinaceae exhibits different habits. In fact, it grows in different habitat also. It has small annual herbs that grows like ascending to erect or the procumbent or spreading, as well as it has perennial herbs that you will get the flowers throughout year and the plants will remain throughout year. For example, our home plants, Brassica. Similarly, it has a climbers. It has a straggling climbers like a septolerion, or some of the Commelinaceae also grows straggling. It has a diversity in the inflorescence also. If you take an example, if you have the Brassica in your hand, it has a spathious bract. Similarly, Commelina bengalensis, it also has inflorescence of the flowers enclosed in the spathious bract. If you see some of the Mudanias where the terminal panicles are there, we can call it as a thyrsis. In fact, some flowers are born in axil of the leaves. Some are having like a leaf opposite fascicles, and some are having a globose kind of inflorescence. So this is entire diversity in the plant family, Commelinaceae. See the diversity in the flower color and shape of the flower. In fact, the arrangement of the flower organs. I will go by one. The Commelina typically shows the typical two lateral petals, that is what showy petals, and having the showy antherodes. What you are finding over as yellow colored dots are the antherodes. In fact, as I told you, the Taxonomy and the cynotes is having 6 fertile stamens and moniliform hairs. Hairs, that is what one of the unique characters belongs to the genus called cynotes. And the inflorescence is also like a sorry, I may be having the poor connection, but I'll come to the last slide. So no one can make the differences between the sepals and petals over here. All are looking like a similar. The outer wall we call it as a sepal and the inner wall is a petal, but having the uniform color. And the Rhophalophora, which has a unique sigmoid kind of stamens, and the symmetrical petals of the Mudania, striated petals of the Mudania, and asymmetrical or the bilateral symmetrical petals of the Mudania. So this is what the diversity we are having in a single family called Commelinaceae. And it is very fascinating to find out the arrangement of the different organs. So if you can see the flower of Dictyospermum, the wall of scent, but the ovary or the pistil is diverted outside to the stamens. It means that the enantiomer style is also observed. Here, 3 unique staminodes you can make it out. The lemon yellow colored staminodes are there, and 2 lateral stamens are there. Here, the medial stamen is rudimentary. So, this is very unique feature of the certain genera where they produce the Cleistogamous flowers. I'll come to that point, and I recently read that those plants produces the Cleistogamy, usually having the asymmetrical petals, and it evolved almost 160 times. So you can make it out that how they have been evolved. And I'll come quickly to the shape of capsule and the dehiscence and number of seeds. As I discussed, the capsule is typically trilocular, but it is not uniform throughout the family. Unilocular capsules are also known in the family. Bilocular is also common, and more common is trilocular. Here, in the capsule of Dictyospermum, 3 walls you can make it, 1, 2, and 3, and every wall has a single seed. So it's the remittance of the seed over here. But in case of Mudania, seeds are arranged biseriately in a row of 2. So this is one, this is another. So likewise, trilocular capsule and biseriate seeds. So this is what the usual terminology we are using. One unique character of the genus called Polia, which has indehiscent capsule. It's often difficult to call them as a capsule. So we can call it as a becoid fruit or the fruit like a berry like a fruit. But even if I call it as a berry, we have certain limitations because it is a dry. So dry berry or the dry becoid fruit we can call. And here, see the shape of the capsule and how the seed nested in it. It looks like the bird's eggs nested over in the nest. So this is what the uniqueness, and elysomes are there in the seeds, appendages, we call it as the waxy appendages over there, and that plays the role in the dispersal. For example, many of the ants carries those seeds, and the dispersal that happens, that provides the foods to the ants. And sticky hairs, I don't know whether you can make it out, the hairs over there, the glandular hairs of the Rofalophora that we call as Anilima now, that usually dehisced or the sprayed by the animals, that the mammalian sprayed that we have recorded in the Rofalophora. And of course, likewise the habit, inflorescence, flowers, capsules, we are having tremendous variations in the seeds. So this is Commelinaceae, this is Cynoides, this is Anilima, this is Mudania, and this is both dorsal and the ventral surface. Here you can make out the attachment to the capsule that we call as a hilum. This is embryotega. This is embryotega. So position of the embryotega means a lot, and depends on the position of the embryotega, you can classify the genera. So this is the uniqueness that the seeds of the Commelinaceae has. I'll go quickly. The genera. This is Amiscotolite. Earlier it used to be called as a Forestia, and we have 3 species in India. Recently, another species that has been added from the Andaman and Nicobar Islands. Total 26 species are there in the genus, and uniqueness of this group is having the leaf opposite sessile fascicles. It means the flowers are sessile and they are clustered together, forming a globose kind of inflorescence. So this is the typical characteristic of the Amiscotolite. And succulent sepals, another character of it is that it has like a succulent floral organs. If you take the petal or the sepals, both the organs are succulent. So we have a glabrata, dolichondra, hookery, and monosperma. This is the monosperma recently added from the Andaman and Nicobar Island. It grows ascending in the forest, undergrowth of the forest, and has a many number of flowers. As compared to this species, the Molissima glabrata, this has a multiple number of flowers, and this is known from the Andaman Nicobar Islands and the Southeast Asia. I forgot to mention that it has some epiphytic taxa also. For example, the genus called Bellocinopsis, the species particularly Vivipara, grows on the tree trunk, moist tree trunk in wet evergreen forest. And this plant belongs to the tribe Tradescansi because it has a six fertile stamens. If you see the plant growing on the tree trunk, and similarly another species Bellocinopsis cuensis, which is in now in European botanical gardens as because of its ornamental potential. See both the surfaces of the leaves. One has a dark green, other one has a vinaceous or the dark purple leaf surface. So this become very popular, and recently it was rediscovered from the Tirunelveli, Tamil Nadu, and this you can use it as this plant has a typical ornamental domestication potentials. So you can use this plant as in hanging baskets or as an in-house plant. So this is very unique plant, and because of it is endemic, it is not much popular. This is a beautiful illustration made by the Bedome in Bedome's icon, where he had showed clearly the dimorphic flower leaves color. And this, few of you know this genus called Calicia. We have many garden ornamentals. This is the Mexican originated, but now widely spread throughout the world. And it is also popular as the Tradescantia is. Now I'll come towards one of the diverse group, Commelina, where all around 170 species are there. It is more diversified in Africa. We have around 28 species, 27 species, 27 to 28, and out of that 6 are endemic. So these are the endemic I have listed. If you see the habit, it is very diverse. For example, if you take an example of the Commelina badamica, you will get the erect, typical small erect habit. In fact, the Commelina andamanica, if you see, that has a typical spreading habitat. You will get the mat on the floor, and in fact, in your area, you will find the Commelina attenuata that also grows very procumbent to the soil and with having the typical azure blue flower. So flowers, if you go to the field in the early morning, then you will be able to see the flowers. Otherwise, it is highly difficult. Then I will come towards the Cleistogamy. As we know, almost we are acquainted with the chasmogamous or the aerial flowers. Commelinaceae has the Cleistogamous flowers. Don't mess up with the roots. These are not roots. These are like aerial shoots turning down, like in Arachis hypogaea in groundnut, and there the self-pollination happens, and the capsule formation used to be happen. And this is what the Cleistogamous flowers usually occurs in Commelina bengalensis, Commelina forscale, and Commelina. One species is there, Commelina, that is very endemic. I will come to know. And this is another Commelina which has the typical leaf sheath and ligulate leaves. Here is the ligule, and these are the typical bract or the spatulaceous bract of the Commelina, where you will find different syncynies or the different type inflorescence grouped together. And here the flowers are borne in axillary with long peduncle. Here flowers born in the terminal but with the short peduncle. Here also the similar situation. Now I will come towards the space diversity. As I was telling, the space is very unique. Space is not fused at margin in many species of the Commelina, where in this case, these two plants or these two species, Commelina curzi and the Commelina bengalensis, spathe is fused at margin and it is arranged in terminal in a solitary or in the aggregate forms. It means that number of spathes are aggregating together. So I have dissected the spathe to find out what exactly, how exactly the flowers and the inflorescence looks like. So I found in some of the species, the upper cincin is completely missing, where the lower cincin produces the male as well as the female flower. So here is the one of the open flower, and here is the preceding flower which turned into the capsule. Here the similar situation. The capsule formation happened. There is no sign of any upper cincinus. It means another branch of the inflorescence. Only one branch is there. Similarly, here you can make it out. One branch is there, and this is the lower cincin. So this is I wanted to express over here the free margin of the spathe, and here I have dissected. I have mechanically teared off the spathe. So I have a puzzle for you people. You may have seen the flowers of Commelina forscalae. So this is what I have presented over here. Two flowers are there. I hope the participant can view the flowers properly. Here, the difference between two flowers, anyone can make it out? The difference between the two flowers? It is so difficult to explain it virtually. But if I have a physical lecture, I mean to say lecture in presence, then I can ask you to raise your hands and tell me what is the difference between two flowers. So this is the flower of upper cincin, and this is the flower of the lower cincin. Upper cincin, if you see, this part is missing. It means this flower is a sterile flower, or the male flower we can call. And this is the fertile flower or the bisexual flower, where the pistil is there, two stamens are there, staminodes are there. Here, that one pistil layer is missing. So this is what the uniqueness you need to find out. You need to have a keen observation in the field, in the flowers of Commelina or in Mudani also, you will find there are certain differences. That is what to accommodate cross-pollination. That is only because of to attract the pollinators. And I don't know why the plants are wasting the energy if they are having the Cleistogamous flowers as well as the aerial flowers. But definitely we'll find the answer in the future. So whenever you are in the field, you need to have a clean observation whether the flower is fertile or sterile, or the male or the bisexual or the hermaphrodite. So see the color variation. You have a lilac, you have a blue, dark blue, and you have a ochre yellow. And the arrangement of the petals, if you see, that is very unique to the every species. So therefore, the variation is kind of a key character. You can use the number of spathe, fusion of the spathe, number of capsules, number of flower color, number of seeds. In fact, petal shape also can be a good character. So these are the different characters you can use in the field. And one thing you should notice, if someone wanted to work to explore the Commelinaceae flower, you have to be in the field as early as possible. As I said, flowers are, we call it as a dew flower or the day flower. They bloom at very particular time of the day in the morning, and they will fade down by the afternoon. So if you are there, I remember one incident. I went to Tirupati hills long back in search of some Commelinas, and I was unfortunate. I didn't went to the temple to get the darshan of Lord Tirupati Balaji, because I have to click the flowers in the field in the morning. So I skipped the plan to visit the temple, and I spent the entire morning with the family Commelinaceae or the plants Commelina plants. And when I returned back to home, I mean to say my hometown, my friends asked that why didn't you went to the Tirupati? I said I'm very hurried because I want to click some Commelina photographs, and that's why I missed the darshana. So some incident happened. So you have to be very alert in the field when you are clicking typical Commelinaceae flower. Because if I go for darshana, I may have missed the opportunity to click the flowers. Of course, laterwards I went to the temple with many of my Andhra friends. So that was the incident I wanted to share. And you have to be very clear while taking the photograph. Because one view or the one-sided click will not enough. You have to have a multiple click. From the front view, from the side view, you have to open the space. You have to see the branches of inflorescence. So all the things, and it is like not on a, it's not a flat object. So you have to be very particular with the focus also. Because if you are focusing the stamens, maybe the other words are getting out of focus. So this is what you have to be very careful. You have to be in the field early in the morning. Second, you have to be very careful while clicking the photographs. And this is how, if someone sends a photograph for identification to me, I used to ask, I need a more character. It is not enough that you have a single photograph of the great looking Commelina will not going to be enough to identify the plants in the field. So I request all of you, those who are interested to learn some Commelinaceae and those who are want to be acquainted to click the photographs of the Commelinaceae, be sure that you are in the field early in the morning. And secondly, you should focus and dissect all the parts in the field itself, because flowers are very, very delicate. You cannot press it. If you are pressing, then it is difficult to re-dissect all, because I found very difficulty to dissect the flowers from the pressed specimens. So I am taking care whenever I am in the field. I used to click the photographs properly. So this is number of space, number of fruits also you need to observe. Here we have added this plant to Flora of India, Commelina imberbis, where you can see the number of fruits. Almost 7 or 8 fruits are there, capsules are there per spade. And in fact, Upper Cincinnati also having the capsules. It means that it produces Upper cincinus with the male flowers. So I will come towards the cynotes. Now, likewise, the Commelina, cynotes is diversified. Because if you compare the number of species, maybe most of the species are showing the polyploidy, and because of the ploidy nature, many of the species are morphotypes or the cytotypes. So few species are endemic to India. Total, we are having around 15 species. In entire world, we are having 56 species. See how small the plants are. This is Cynotes tuberosa. I hold it in the hands and see the flowers and see how big. It is not, I mean to say, accommodating in a single frame. So Cynotes villosa over here. So the drastic diversity is there. If you see, the flowers are enclosed in the small bracts like a falcate bracts, and over here, no such bracts are there. The leaves itself turned as a bract, and these are the axillary flowers. Many of you have seen these flowers of the plant. This is what the Cynotes axillaris. So therefore, this is very unique. Tell me the difference between the two flowers. Your right hand side, I have posted one flower. On the left hand side, one more flower is there. You see the color of the stamens and the color of the filament hairs. In right hand side flower filament hairs are like a paint white, and over here is the entire, it's like a unicolor or the uniformly blue. And stamen is also in yellow color. Over here, yellow color is like very rare. These are like black color, more often black color. And that is how taxonomists have identified these species. This and this are distinct from each other. So one plant is Archotensis, Cynotes archotensis, where the other is Cynotes hispida. So the Geospermum is one of the Commelinaceae member, where 3 species we are having, and it has a typical inflorescence, and the flowers also, as I told in my initial slides, flowers, petals and sepals you cannot distinguish. So this is having the sepaloid petals and the petaloid sepals. So 3 species we are having. One is known from the Andaman Islands, while other 2 occurs in the Western Ghats. So see, some eyed flowers. What does it mean? It means that pistil is locating below, and the stamens are covering. I mean to say, stamens and the staminodes are shading from the above. It means that it is staminal wall is covered entirely towards the pistil. So this is what the thrum-eyed flowers and Floscoba. This occurs along the streams. It's like a perennial plant, and it is unique because it has a glandular sepals. Mudania, one of my favorite, as I told in the when we started, I showed my hand with the Mudania striatipetalaraki. So total 50 species known from Africa and Indo-Malaysia, where we are having the, I mean to say, India is home for almost half of the species, around 28 species that we are having, and we have a good endemism. See the diversity within the flower color, flower shape, and the problem is whenever you are looking towards the specimens, as I told you, the flower is such a delicate, you cannot dissect it. For example, this is Mudania esculenta, and you cannot differentiate it from the dimorpha from the esculenta, because in absence of flower, it is very difficult, highly difficult to isolate both the species. So live as well as the field observations as well as the herbarium observations are both are equally helpful to identify such complicated genus species. We have done the scanning electron microscopy. As I told, the position of the embryo taga is very unique to the particular group, and this is how the Mudania has a diversity in positioning the embryo taga and positioning the hilum. Similarly, like a dictyospermum, we have a polia, and it also has a good diversity in India. Almost 5 species we are having, and mainly restricted into the northern eastern states of India. In Peninsula, we have 1 species called Polia subumbellata, and one more genera we have added to India, that is Porandra. We have seen the Amiscotolite. Some connections problem. Yeah, I will come back to the last slide. I was here. So I will come towards the Streptolirion. As I said to you, that Streptolirion is one of the interesting genus in the family Commelinaceae, as it has spreading or climbing habit. It means that I have drawn here the Streptolirion, and you can make it out the habitat of the Streptolirion. It has the typical cordate leaves, and inflorescence also enclosed in the spade, but it is not like in Commelina. it has a much branched space inflorescence. For example, here is the lower cinciny, here is the lower cinciny, one more cinciny is there. So like composed of multiple cincines. And in addition to the spathaceous bract, the flower bracts are also unique. So this is the very unique genus to the Commelinaceae, and we have two taxa. One is Streptyluro volubile, and other one known from the Khasi hills. We call it as a subspecies Khasianum, which has the rufous pubescent hairs. This is Tinantia. It is introduced the plant now naturalized on the Dehradun Mussoorie Road, and total 13 species are there across the world, mainly in Mexico. And you are very aware with this group called Tardescantia, Calicia, and earlier we used to call it as a Riho, Setcracia, and rest. This is one unique genus of Commelinaceae called Tricarpellema. It has, of course, many of the plants are having the 3 capsule, but this is very unique because other rest of the genera are having trilocular capsules and number of seeds are very limited. But here, in some of the Tricarpellema species, number of seeds are very high. So I have certain questions that to the developmental biologist, so that how these plants are evolved. For example, how they have diversified. We have a different species of Commelina, Tricarpellema, Floscopa, Cynotis, Mudania, Dictyospermum. But how they have evolved and how they have, I mean to say, adapted to the certain conditions. So Tricarpellema is almost looking like a Rhophalopora, but it also again unique. That is the whorl leaves are there, then the inflorescence is completely covered with the glandular hairs, then the flower color is typical. In fact, the capsule is also typical. Then the Rhophalopora has been diversified, or the how speciation happened in the group. That is very interesting to find out. So in the almost a decade we are working, and in this decade we have contributed some important species to the family Commelinaceae. In 2009 we have described one plant from the Maharashtra that belongs to group called Mudania, and this is very unique because of its fleshy red flowers. We named it as a brown eye. And the other one with Dr. Venkataramana, we have described one species from Andaman and Nicobar Islands that we named as a saddle picensis because it is restricted in the Saddle Peak Islands. And similarly, we have described one new species in Commelina from Badami Plateau. We named it as a Badamica. And from Maharashtra, again, we have described one more Mudania, which is very similar to this Mudania striatipetala. But of course, it is different because in the Mudania striatipetala, you may find the orbicular petals, but here in the petals shape is quite unique. And here again, we have described one Commelina species in 2018. We named it as a rupestris because you know what, it occurs in the rocky habitat of the dryland forest of Karnataka, and it grows amongst the rocky crevices. Therefore, we call it as a rupestris. And this is very similar to the species called Commelina hispida, but it is very unique to the Commelina hispida as it has a smaller, much larger flower than the typical Commelina. And similarly, one more species we described from the Andaman Nicobar Island, from the Chidia Tapu of the Andaman Islands, that is with the MC Naik from the Andhra Pradesh. And we named it as the Litoralis. The top species we have named rupestris because it occurs in the rocky habitat. The down species we named as a Litoralis as it occurs in the littoral habitat. It is different from the other species called diffusa and caroliniana with different characters. And similarly, we also worked on the rediscovering the plants. This is Mudania striatipetala. We have recorded from the coast of Chennai, and this is very, I mean to say, eye-catching plant. And there was a story that one of the enthusiastic photographer, now she is settled in China. She took the photograph as a curiosity, and she was no idea what it is. So she sent this photograph to the Tabish Qureshi, who is like a founder of the Flowers of India. Many of you are aware. And from Tabish, it came to me to identify. I got surprised because I never saw such plant from India. So I recorded this as a new record. Then I went to Chennai. I found this plant growing abundantly over there, and this is kind of a rediscovery that we have made after long years. And similarly, in last visit of Andaman, we have collected one plant that was not collected for almost 150 years, that we collected, and we have added to Indian flora. Likewise, we found one African plant in the Belgaum district that identified as a Commelinaceae imberbis. That is earlier it was known from Africa only. Now it is also occur in India too. And this is the small journey. I believe the 1 hour is not enough to express all the things over here. But I tried to convey all the possibilities that to do the further research. I believe that developmental biologists or the people who are working in the evolutionary or in ecology should study this family very critically, because being an expert in the taxonomy, I have certain limitations to work on its developmental biologist. But I believe that there is a lot of scope to do the further work. The certain questions are there. As I said, why enough number? For example, Comella bengalensis, number of plants are there. It's like a weed everywhere in the fields, around the roadside, along in the ditches, everywhere undergrowth, everywhere it grows. So, what is the need to produce the Cleistogamous flowers? As they are producing the typical aerial flowers and having the good seed number also. But why they are like producing Cleistogamous flowers? Similarly, why the enuntio style is there, the curvature of the pistil is there that is of course to promote the pollination. But in what way? In fact, the range of this petal variation or the sepal variation or the inflorescence variation, why it happened? Even I didn't talk about the cytological variations. That is also a very unique feature to the Commelinaceae. We have a basic chromosome number 6, and the N, that is the haploid chromosome number 6 to almost 70. So it is quite range of variation. In Cynotis tuberosa, you may find, if you go to the Gingy Hills, you will find the triploidy form of it. If you go to the Horsley Hills, you may get some different form of it. So there is a cytological variations within the family. So entire family, if the collective efforts have been done, the family can be treated better. As an example, I have given the family is nested between the Poaceae and the Gingiberaceae. It means that it is under the continuous evolution, and you may find the unilocular to the multilocular, trilocular capsules with multi-seed, and somewhere the anthrodes are present, somewhere the stamens are missing, somewhere only produces the male flowers. So all the questions need to be answered. I believe that my entire life will not enough to answer all the questions. So it is up to you now. That I hope that this webinar is attended by almost around 100 people. So at least 1 or 2 people can take this family further to understand that evolution is significant. That will be really great. So once again, I am very thankful. It is quite different experience. No, I cannot see the responses from the audience. So this is what I feel very, we can call it as embarrassing, that I don't know what is the reaction from the public. So I am just, it's like a talk. I'm in front of screen. I'm talking to entire family Commelinaceae in a little. As I say, the 1 hour is not enough, but I am very thankful to the organizers that they gave me an opportunity to present my work. Secondly, tomorrow again I'm going to spread about the how artificially to make the taxonomy better. So with this, I'll take off right now and I'll give the screen to the organizers. Thank you.
Thank you, sir. Thank you very much.
Thank you so much, sir. That's excellent presentation. Whole thing presented about the Commelinaceae. I think audience fortunate enough to have a speaker like you. They got much information about the Commelinaceae. And over to Sadhana.
Yes, sir. Am I audible, sir?
Yeah.
Yes.
You are audible.
Yes, sir. So I just wanted to give a word of thanks to you. The way you started.
Before that, any participants would like to?
Yeah, coming to the question, sir. Okay, sir. I'll go on.
Anybody would like to interact? Ask them to raise the hand so that.
Yes, sir.
Yes,
there's one question, sir. What are the applied aspects of Commelinaceae?
So one person I'm going to.
Sorry, sir. Mayur, sir. Shall I repeat the question?
We lost his signal. Let's wait. Meanwhile, the participants who want to ask questions, they can raise their hands so that we'll allow you to the panel. Once the sir joins us, you can ask the questions. Or otherwise, we will share the contact details of sir so that you can pose your questions directly in mail.
Ornamental potentials they are important because of their ornamental typical foliage. Many are like a cultivated. In fact, as I told you, the Beliocinopsis hello, am I audible?
Yes, sir. Yes, sir. You're audible, sir.
Hello.
Sir, you're audible, sir.
So many plants that can be domesticated. Yeah. Many plants of the Commelinaceae can be domesticated, and these are having great ornamental potential. In fact, if you come to our garden, you will find we have domesticated 3-4 cyanotes. In fact, the plants that I have rediscovered in our premises. So, in fact, one of the plants that I bought it from the Uti, and it is growing under the coconut farm like anything, and it is like other weeds are not coming up because of that plant. You can play with the plants if you want it to be, but there are less as rightly associated with the mankind. And those who have a Commelinaceae plant, those can write to me. My mail is like I have shared with the organizers. In fact, you can write it to my mail ID. I'm writing my mail ID over here. So you can write to me if you have some doubts, some clarification, or you have some unidentified tiny dew flowers. I'll be really happy to help. And there is a pollination mechanism for the Commelinaceae. There are several agents that we have found. The bumblebees are there, then the different. I have seen the wasps also visiting the Commelina flowers, but in some genera like Rofalophora and the Poolia, self-pollination, if it has been not rewarded by the pollinators, plants itself has advantage to have a self-pollination. The calling of the stamens and the other fertile organs will start once the flower going to be faded. Pollination is also triggered, and there are some biology of the Commelina, and it has been well studied by Dr. Fadden and Hunt. And there is again the pollination mechanism is like a kind of evolutionary factors, and it is under the continuous evolution. For example, in Aneuploidy, as I said, in Aneuploidy occurs in the genera called Mudania in some species of Commelina and Rofalophora. But it is very interesting to find if you have some observations particular on the pollinations, and also it is difficult to identify whether the particular pollinator is visiting the flower to pollinate the flower or to as a pollinator or as a cheater. That is cause Commelina flowers, no, many of like get cheated because of it has a colorful, attractive staminodes rather than stamens.
So shall we go with the next question?
No, share signal is not available, right? Sir, over to Vijay Bhaskar, sir.
Hello.
Sir might be rejoining, sir. Meanwhile, we can.
Organize.
Dr. S. Vijaya
Sir
would
has
like
come,
to
sir.
interact.
Come,
To
Sir Mayur sir, come.
Yeah, please.
So connectivity is very poor over here.
Yes.
I hope that the lecture interrupted in between many of got an idea that what exactly I wanted to convey.
Dr. Vijaya would like to interact. Please.
Madam, you can unmute your mic and speak, madam. Dr. S. Vijaya. Madam, I just raised your hand. Sir, no, she said.
Parmesh, Mr. Parmesh would like to ask something. Mr. Parmesh, please raise your hand so that we can connect you.
Hello.
Sir, please pose your question, sir.
Sir, may I audible?
Yes, yes.
Sir.
Hello.
So you're audible, sir. Please go on with your question.
Yes, you are audible. Please proceed.
Sir, is there any medicinal importance of Commelinaceae, sir? Plants. Hello, sir.
Yeah,
Yes, sir,
please wait. Sir will respond. There is poor connectivity.
Yes, sir. Please.
So I was out for the question. I don't know what Parmesh asked.
The pharmacological importance of Commelina. What is the pharmacological importance, medicinal importance of Commelina?
That is what I answered. No, there are very less. There are less pharmacological. But you can read, Parmesh, that to understand more about the pharmacology. I will be happy to help in this regard because we work especially on the taxonomy part. Recently, we have one article that is what I told you on salicylaldehyde content of these endemic plants. So I believe that because all these commelinaceae having a
Sir, some interruption is there from your side.
Vijayabhaskar sir, can we conclude the session, sir?
Yes, yes. Yeah. When I approached sir, Dr. Nandika sir, immediately readily accepted for the session, not one session, for 2 sessions, yeah, he accepted. And excellent sessions we have. One already I have seen, and tomorrow again he will join with us. And if you have any doubts regarding this, you can pose questions tomorrow also. You can interact tomorrow also, no problem. Now I request Sadhana to propose vote of thanks.
Yes, sir. So Mayur sir will be adding, or shall I?
Yes, you can.
Continue.
Okay. The way Mayur sir started, like he was connecting all the subject with the personal experiences he had, like all the way from the starting, like how and why he chose particularly Commelina for doing a research or working on it. So sir was connecting all the subject with the personal experiences he had. That was very helpful, and maybe it will be more helpful to people who aspire to work on the subject, work on any subject further. And mostly we get to see PPTs which are more flooded with the content and a lot of information. But PPTs, Mayur sir's PPTs were like one picture and one word, and the pictures were so clear that we did not feel it was like a virtual session. We literally felt like we were walking around some garden or some place and we were listening to the class. So the pictures were really clear, and we had many messages in the chat box too about the pictures, that they were very effective. And also the explanation sir gave about every minute detail about Commelina, like maybe the flowers, leaves, and all the aspects were covered very clearly. And as our sir gave an introduction of 5 minutes about Mayur sir, and literally at the end he said like it was a brief introduction. And having a person who has a brief introduction of 5 minutes and so many accomplishments in his career, uh, having such person in our session is a privilege, and we are highly privileged to have you on this session, sir. It was very helpful for us and very informative for us. Thank you so much for being with us, sir.
Yeah,
Thank you. Thank you, Sadhana ji. Thank you, Dr. Reddy, for calling me over here. So
Yeah.
probably tomorrow we will meet on the time
Sure,
for the other session. Thank you.
Thank you, sir.
Thank you, sir. Thank you very much, sir.
Next speaker is with us, Dr. S. Karnaka Reddy. Yeah, while structuring the course, taxonomy, plant taxonomy, we added many things, and we missed one cytotaxonomy. We approached many persons like Dr. Manoj and other people. They were all busy, and we could not find a suitable person for this. And finally, we approached Dr. S. Karnaka Reddy regarding this cytotaxonomy. Now he's not having any research in that, but he's good at cytogenetics, and he immediately accepted our request, and he came out with the cytotaxonomy. So now I request Dr. Shashikala to introduce Dr. S. Karnakar Reddy.
Sir, and thank you very much, sir, to give this opportunity to me, sir. I take the privilege to introduce today's our eminent resource person, Dr. S. Karnakar Reddy. He is assistant professor from the Department of Botany, University College of Sciences, Usmania University. His educational qualifications are he did a BSc from Usmania University with first division, and did MSc Botany from Usmania University again in first division, and he finished PhD from Usmania University in 1990, and afterwards in 2002 he did advanced PG diploma course in bioinformatics. He in his tenure, he attended different refresher and orientation courses, and he has got good teaching experience. He initially started his teaching as an ad hoc lecturer, and finally he joined with the Usmania University as a permanent faculty member. He did his postdoctoral research from 1993 to 98 as a research associated from UGC. He had a fellowship from UGC as JRF, SRF, and research associate. He has got very good technical knowledge in molecular modeling and tissue culture studies. He had also computer knowledge, especially C, C++, Java, and Oracle. He handled different research projects, different minor and major research projects, especially which are funded by UGC UP FAR program. From 2014 to 2020, and he also got a UGC MRP project also in the year 2011 to 13, and then he also got UGC minor research project which is funded by CIRO in 2008 to 2010. He was invited to give extension lectures by different colleges and universities. To his credibility, he finished 6 PhD students, 6 PhDs from his laboratory, and 6 more students are working under him. He has got a membership, professional membership in different professional bodies like Indian Science Congress, and he published nearly 26 scientific journals. He did different administrative positions, worked as warden for PG hostels and coordinator for environmental sciences from 15 to 17, and worked as warden for new PG hostel from 19 to 20. So this is very brief about Dr. S. Karnakar Reddy, sir. We are very, very happy to have you here, sir, as a resource person. Now, this is very brief about you, sir. Now you can take the session and you can enlighten our participants, sir. Thank you very much, sir.
Yeah, thank you, ma'am. Now we request Dr. Karnakar Reddy sir to share his screen and start the session.
Madam, kindly mute your mic, madam. There is some disturbance from your side.
Sir, please unmute yourself. Dr. Kannakared, sir, please unmute yourself and start. Dr. Kannakared, sir, please unmute yourself.
Sir Karnaka sir, on your screen you'll be seeing one sharing tab,
If it
sir,
is
green
okay?
color tab. Yes, sir. Okay, sir.
Yes, sir.
Uh,
You can start.
Good afternoon, all of you. My special thanks to Professor Vijay Bhaskar Reddy who organizing this session, and my special thanks to Shashikala Madam. She explained very nice manner, so it is better than my lecture what I am feeling. Today I will like to speak on cytotaxonomy. So myself, Dr. S. Karnaka Reddy, Assistant Professor from Department of Botany. So the cytotaxonomy, which deals with the any plant species in the form of the chromosome number and chromosome size and the chromosome behavior during meiosis. So in this chromosome number, any plant species having the specific chromosome number. For example, you are seeing the pea plant that is containing the 7 chromosomes, and in case of the onion, we can see the 8 chromosomes. Like that, we can see the different plant species may have the different chromosome number. The chromosome number which gives the identity for a specific plant. And also we can see the size of the chromosome. The chromosome size maybe varies within the species from plant to plant, we can see. And also we can see the chromosome behavior during meiosis. It indicates whether they are same species or two different species. So taxonomy in relation to cytology. So the cytological characters of the chromosome number, morphology, and chromosome behavior during meiosis. So number, we already know that each plant species having the specific chromosome number, and we can see the number changes from within the same genera in different species. We can see the different number of chromosomes. So the morphology of the chromosome, while we're taking into the chromosome which is having the 3 different regions, maybe in the form of the centromere and 2 arms. In the form of the short arm, we generally call as P arm, and long arm as a Q arm. So it is connected in between in the form of the centromere. So on molecular basis, we can see in the form of the telomere in addition to centromere. It keeps the chromosome length properly. And also we can see another center, that is the replication center, which is useful to chromosome duplication. So like this, we can see the morphology of the chromosomes in the taxonomy. There are 3 or 4 types of the morphological views we can see in the form of the V-shaped chromosomes and in the form of the L metacentric chromosomes when the P by Q ratio is equal to 1. So when the P by Q ratio is somewhat less than 1, we can classify into submetacentric L-shaped chromosomes, and in the form of the acrocentric, in the form of the J-shaped chromosomes, and in the form of the telocentric, in the form of the I-shaped chromosomes. And like this, we can see in the form of the chromosomes in the different way morphology. So like this, we can see the different forms of the chromosomes in the form of the 4 types. In the meiosis, we can see the chromosomes pairing maybe differs from the homologous chromosomes in the non-homologous chromosomes when we see in case of the F1 hybrid. So this gives provide an idea of the genetic relationship between the plant populations in the chromosome number. So this is the one of the chromosome slide. We can see the in the form of the different chromosomes. So what is karyotype? How to see the karyotype? We can see uh, karyotype can be defined uh, as the phenotypic appearance of somatic chromosomes in contrast to their genic order. So most of the taxonomists will look into the morphology and chromosome number, whereas cytogenetics, which look into the genic order. Sometimes the chromosome is same in the form of the size and shape. But when you see this, when they are paired in the meiosis, we can see the genic order. For example, in one of the arm, we can see the gene sequence in the form of ABC. So that will be turned into CBA by means of the inversions. The inversions, you know that there are two types in the form of the paracentric and pericentric. So when the inversion which is taking place outside the centromere, that is known as the paracentric, and we can see sometimes the homologous regions will come together and pair. So that is what we can define karyotype, defined as a phenotypic appearance of the somatic chromosomes in contrast to their genic order. So this mostly the real taxonomists won't bother about the genic order. They will look into the morphology and size. So here the karyotypes, we can see the chromosome number. It is gives the how many number of chromosomes are there, and also size, and in addition to the form of the chromosome. So the next, it is the C mitosis, onion root tips, where you can see the number of chromosomes. You can count it very clearly under the microscope. When you treat with the colchicine, the chromosomes will be spread like in a box. All the chromosomes, you can see their number, their size, and also form, and it gives the very interesting feature. If you take the root tips and you can fix it, particularly we did in the onion root tips when I was working in the cytogenetics in the Cypher Science College. We used to study the chromosome morphology, and we can see the different karyotypes. So if Rio discolor, we can see the but in that plant we can see the perfect V-shaped chromosomes. Here we can see in the onion root tip, we can see the submetacentric and metacentric chromosomes. So what is C mitosis? Actually, the colchicine treated mitosis. We can treat the onion root tips with colchicine, 0.5% solution for 1 hour. The root tips, then root tips can be washed with the distilled water. Root tips can be fixed with 3:1 ethanol and glacial acetic acid. And after that, we can squash it. We can see the what type of chromosomes are there, how many number of chromosomes are there. That gives the immense knowledge in different plant species. So whatever the karyotypes, we can arrange in the form of the ideograms. So ideogram is a diagrammatic representation of the karyotype. So in the form of bar diagram and ordered in a series of decreasing the size. So we can take the karyotypes, whether it is normal or the symmetric or asymmetric based on the ideogram. So we can arrange the chromosomes in the form of the decreasing order and metacentric to submetacentric and acrocentric like that. So that is what ideogram. So we can see the onion root tips. If you construct the karyotypes in the form of the bar diagram, here you can see that there are 8 chromosomes, 1 to 8, generally in the decreasing order, and we can see the metacentric to submetacentric. So this is the symmetric karyotype. Maybe this type of karyotype may be considered as a primitive karyotype. So like this, we can see the one of the symmetric type of the karyotype. So in addition to that, we can also see the asymmetric karyotypes in case of the Vicia faba. So in the Vicia faba, we can see the there are 6 pairs of the chromosomes are there. So in the karyotypes, we generally take into n number of the chromosomes rather than the 2n number of the chromosomes, and we can arrange the decreasing order of the sites. So here, this is one of the standard type of the karyotype in the asymmetric type of the karyotype. So you can see the centromeres and also secondary constructs and in the form of the satellites. Generally, we can see the centromere in the primary construct. In addition, we can also see the secondary construct. So here we can see it is considered as a nucleolar organizer, and we can see the a dot-like structure on the top, maybe in the form of the satellites. So sometimes we can see the more secondary constructs in case of the whenever the diploids, we can expect to see the only one secondary construct. In case of the tetraploids, we can see the 2 secondary construct. Based on that, we can also define the how much ploidy is there in a particular species based on the number of secondary constructs. So like this, the karyotypes we can see in the form of the symmetric and the asymmetric type. The symmetric types may be in the form of the considered as a primitive type of the karyotypes, whereas asymmetric types may be in the form of the evolved type of the karyotypes. So based on chromosome number is constant, same in all the individual species. So you can see the any species having the certain basic number of the chromosomes. The chromosome number may be constant. It gives the certain conformity of a property distinguish a species from the member of the other species. For example, if you see the chromosome number in the human beings only 23 pairs. So, but some extent we can see the each plant species, wheat is equal to 7 chromosome number, the basic number, and we can see in the form of tetraploids and in the form of the hexaploids. So like this, the chromosome number may be specific plant species may be very specific. So based on that, we can identify the how many number of chromosomes are there in a specific plant, and we can identify. So generally, in the taxonomy, the chromosome number may be useful some extent whenever you are not able to distinguish some extent on the morphological basis. So here is an example, the separation of the species level. So one of the plant genus which is having the 2 different species in the form of the monotropa hypophagia looks like and hypoptitis and hypophagia. These are the 2 different species. In this monotropa genus, hypoptis and hypophagia, they are looks like morphologically similar. But we need to distinguish whether these 2 plants are the same species or 2 different species. And generally we can take in a simple test by taking the chromosome number. Here the hypophysis is having the 48 chromosomes. That is the basic number 8 into 6, 48 chromosomes. So this indicates the 48 chromosomes, and hypophagia is a diploid, 2n equal to 16. So you can also see some of the varieties in the form of the Hibiscus rosa sinensis. If you observe in the gardens, you can see the ploidy levels maybe differs based on the morphology. Some extent you can see the whether they are diploids or tetraploids or the polyploids. So the polyploids generally having thick petals and having the more blooming time. So based on that, you can also see the roses, the cut flowers. You can see the simple petals or thin petals, and the cut roses which is having the thick petals and having the dark color. They are the polyploids. Based on that, morphologically also you can identify whether they are diploids or the polyploids. So here these 2 species are separated based on the chromosome number, hexaploid and the diploid. So hypoptytis is the hexaploid and hypophagia is a diploid. And in addition to that, interspecific separation within the species, we can see the certain variation in the chromosome number at the subspecies level. In case of the Veronica prostata, which is having maybe two varieties in the form of the prostata and the CRA. These two species maybe or subspecies in the form of n equal to haploid number prostata, that is having 8 chromosomes, and series which is having the 16 chromosomes. So like this, these two subspecies are varietal level may be separated based on the chromosome number. So like this, we can interspecific separation we can do in Veronica prostata and Veronica prostata CRE in the form of the chromosome number. In addition to that, we can also separate based on the chromosome number at the generic level. So one of the genera, Cicinidia, is separated into microcala. So in this, we can have the Cicinidia filiformis, actually Cicinidia fusilia, that is converted in the microcala fusilia based on the chromosome number. Here, this Cicinidia genus having the chromosome number in the form of the 13. And whatever the Cicinidia is kept in the microcala in a separate genus, it is having the n equal to 10. So like this, the separation at the generic level in case of the Cicinidia and microcala, basically the microcala within the Cicinidia, it is separated from the Cicinidia based on the chromosome number. So another example in case of the chromosome number based on the genus, in the Tephrosia, most of the species of the family Fabaceae having the 22 chromosomes. But one of the Tephrosia constricta, which is having the 2n equal to 16 chromosomes. So it is separated from the genera Tephrosia into in the form of the sphinctospermum. So like this, these two genera which is separated within the family Fabaceae based on the chromosome number within the same genus, which is converted into Tephrosia and the sphinctospermum. So like this, we can convert in the two genera. In the tribe, one of the Borinae of Rutaceae, the chromosomal base number varies from 7 to 19. So like this, they are arranged. So the members are rearranged with the unique system. So based on the chromosome number in the tribe Borinae, it is segregated 7 to 19 chromosomes. And also we can see the separation at family level. So families are arranged based on the number and size of the chromosomes. So sometimes we can see the number of chromosomes and also size of the chromosomes. So we can see some of the cotton species, the chromosome number is same, but the size is differs. So like this, I will explain later that one. So here is one of the Helboreae and Aneuploidy of Ranunculus. The chromosome number in the form of the 7, 8, and 9 both have the large and the small chromosomes. Maybe lengthwise long chromosomes and the small chromosomes. So they are rearranged based on the chromosome size, based on the number both. In case of the example 2, Ranunculus, n equal to 7, and the small chromosomes are placed in Thylacia, and n equal to 9 and large chromosomes are placed in the Coptoideae. So like this, we can see the chromosomes, small chromosomes, and the long chromosomes. Rather than to speak large chromosomes, we can say size of the chromosome. This is the short chromosome and this is the long chromosome. Like this, in the family Aneuploidy, they are segregated into N7 and small chromosomes, N equal to 9 and the long chromosomes. So this small size chromosome with N equal to 7, they placed in the phylogeny, and N equal to 9 and long chromosomes placed in Coptidae. So like this, we can segregate the number of chromosomes and also size of the chromosomes. In addition to that, the chromosome number and size and morphology, we can also see the chromosome number in the form of the euploids and the aneuploids. So generally, plants, we can see the euploids in the form of a set of chromosomes. So n equal to 7 or 2n equal to 14 or 3n equal to 21. Like that, we can see they are the euploids. In addition to that, we can also see that an addition of the chromosomes in the form of the aneuploids. Here, either addition or the deletion of the chromosomes, we can see it is not a serious consequence in the plants, but it is a very serious consequence in humans. So we can see the particularly in Datura, which is there in the form of the trisomics, but in this slide it is given as a monosomics. So we can see sometimes nullisomics, loss of the two chromosomes, specific chromosomes. In case of the polyploids, we can also examine what is the specific contribution of the specific chromosomes, like in the form of the trisomics or tetrasomics or nullisomics. So, and also we can see the trisomics or tetrasomics in the form of the 2n plus 1, 2n plus 2 like structures. So like this, the trisomics, which are first identified in the Datura stramonium. You know, the Datura stramonium, which is one of the weedy plant, which is having the basic chromosome that is in the form of the 12, n equal to 12. And sometimes in the examinations, they will ask how many trisomics are possible in any plant. Suppose the chromosome number 2n equal to 14. And we can expect either 7 or 14 based on that. Generally, trisomics are equal to the n number of the chromosomes, but not the equal to the 2n number of chromosomes. So here there are 12 capsules are there in the 12 different morphological forms. So each trisomic having the specific type of the capsule. So like this, we can see the Datura stramonium, which is first identified on black sleeve. It is possible 12 trisomics. So in the humans, we can see one of the trisomics, very serious consequence. That is, we can see in the form of the Down syndrome, which is having the 2n plus 21st chromosome. It is in the form of the webbed neck and also mentally somewhat they are not so sound. So based on chromosome number, we can have the karyotypes of the plants is now very useful in classification of the some plants. So generally the monocotyledons have the long chromosome number than the or larger chromosome number than the dicots. Woody plants have the smaller chromosomes than the number. Generally the woody plants chromosomes may be size-wise very less. Generally grasses, you can see the chromosome is lengthy than the dicots. So like that, we can see the each plant species may be having in the based on the dicots and monocots. We can also see the chromosome number variation as well as the size of the chromosome may be varies. Particularly woody plants, we can see the chromosome size is less in length. Smaller chromosome, in short, we can say the short chromosome, which is in the form of the, we can see in the woody plants. And we already explained regarding the shape of the chromosome. The metaphase chromosome is considered as classification. So generally the chromosomes are highly condensed in the metaphase. So out of the 4 phases in the mitosis, prophase, metaphase, anaphase, telophase. So in which phase the chromosome size is less, particularly in case of the metaphase, it is highly condensed, and we can see the shape of number of the chromosomes and shape of the chromosomes and also form of the chromosome. So like that, we can see the while you treat with the colchicine, it inhibits the spindle formation. And also we have the proper spindle formation checkpoint in the cell cycle when a cell is moving from metaphase to anaphase. It needs to form the spindle at all the kinetochore regions. Because of that, we can see the prolongedly in metaphase when you treat with the colchicine, and we can see the colchicine, one of the chemical which is much useful in the plant breeding to increase the chromosome number. If you want to convert to diploid into tetraploid, or you want to produce triploids, you can cross in between tetraploid and the triploid. And like that, it is useful to study the karyotypes in case of the by using the in the form of the uh, chromosome shape, number, and form. It is useful to study the karyotypic studies. And also the position of the centromere and satellites considered as the classification. Generally, we can take the chromosomes or metacentric, submetacentric. Already we explained in the form of the onion karyotype. In the ideogram, we can see the most of the chromosomes metacentric or submetacentric, that is known as the symmetric karyotype. Whereas in case of the Vicia faba, we observed one or two chromosomes metacentric, rest of the chromosomes in the form of the acrocentric or telocentric, that is the asymmetric karyotypes. So based on the karyotypes, we can classify into symmetric and the asymmetric types. The symmetric types may be considered as a primitive type, whereas asymmetric type may be considered as an evolved type. So sometimes the chromosome number may be increased by converting all the metacentric chromosomes, maybe acrocentric chromosomes or telocentric chromosomes. The chromosome number 7 is converted to 14. That doesn't mean the DNA content won't be changed. Like that we can see. So sometimes chromosome number may be increases, but total genome content may be same. And here is a classification of the chromosomes, maybe in the form of the metacentric, when the centromere is at the middle. And we can see the telocentric means terminal, but here in the text somewhat it is not correct. So terminal, the centromere position, that is telocentric. Acrocentric, it is somewhat in the form of the subterminal region, maybe at the near to end, but not the end. Generally, we cannot see any chromosome is acentric. Here is acentric means it is a chromosome fragments, particularly when the chromosome breaks occurs, the acentric fragment will be deleted while in the segregation. So without centromere, the chromosome won't segregate from one cell to other cell. So because of that, the centromere, which is contributes the important thing in the movement of the chromosomes into two different cells. So like this, we have the in the form of metacentric, V-shaped, submetacentric in the form of the L-shaped, and in the form of the telocentric, J-shaped, acrocentric in the form of the I-shape. So like that, we can see the 4 types of the chromosome. Based on the chromosome, so provides the clues about the cause of the sterility. Sometimes we can see the sterility, fertility among the population. And while you want to see the two species, you want to confirm whether they belongs to the same species or two different species. If you cross the two seedlings, and whatever the F1s, you can see whether they are fertile seed, whether they produce the F1 seed or producing the seed or not. So the F1 plants, if they produce seed, and we can say they are the same species. And if you say there is no proper seed production by crossing two different species, and you can see that the seed production won't occur, that indicates they are the two different species. So sometimes you are crossing two plant species or maybe closely related species, and you are diploidizing, and you are getting tetraploids, and you are getting the fertile seeds. So like that, we can see some of the crop models we can see. And also we can see structural difference in the parental chromosomes is the main cause of the sterility. So whenever you see the homologous pairing, so what is meant by homologous? And their size is same and their structure is the same. Means they should have the centromere at the same position, size and structure. In addition to that, I explained the karyotypy, how to define karyotypy, phenotypic appearance of the somatic chromosomes in contrast to their genic order. So whatever the sequence ABC is in one arm, and it is converted in the form of in the inversion, that is in the form of the CBA, or 1 to 3, that is converted in the form of the 3 to 1. So in that case, the pairing won't be occur. So that is what we can see the structural difference, the genic difference. So the taxonomist looking into the structure and size, but whereas the cytogenetics, which will see a genic order of the chromosomes. So when they pair in the meiosis, we can see certain foldings in the chromosomes, and we can see that is not from the same species. In Drosophila melanogaster, there looks like the same, but having the chromosome numbers also same. But you can see the one of the second chromosome mutant. We have the mutants in the form of the structural change, in the form of the deletions, duplications, inversions, and the translocation. That is one of the best examples for the inversions. So that is in the form of the paracentric inversion. So in that case, we can see the sterility. If the two sets of the parental chromosomes are homologous, the plants seem to be fertile. What is meant by homologous? Most of the students do not know what is meant by homologous. Sometimes you can see the 7 pairs in each species, two different species, but you cannot get homology in the F1. But if you make it to tetraploids, you can get the in the form of the two pairs of, for example, A genome and the B genome, and you cross it, that is in the form of the AB in the F1. There is no homology in these AB genomes. And after that, you do the colchicine treatment, and you can do the diploidization, and you can see the A will pair with the A, B will pair with the B, like homologous chromosomes. So homologous means the size of the chromosome and the number of the chromosome generally same, and also structure of the chromosome is same to some extent. And in addition, further depth in the gene order, they should be in the same sequence, even though they are size-wise same and shape-wise same, and but genic order may be different. So that is what we can see the single species. We can see the homologous chromosomes in the form of the two sets. That is what how to define the homologous chromosomes. So here is one karyotype of the human beings. We can nowadays on the molecular basis can give the we have the different types of the techniques in the form of the fish technique, and the yum fish and the gish. So fish technique, fluorescent in situ hybridization, we can give the a specific chromosome, we can find out by the knowing the sequence of the gene of the specific chromosome. So here in this yum fish, so at a time, all the chromosomes are given different colors for the different chromosomes. So young fish, which is given in the form of the X and Y chromosomes and 22 chromosomes. So this is useful In case of the karyotypes, either it is in the form of the trisomics or monosomics or any other things. In the humans, sometimes we have the trisomics, so we can see in this, and also Datura is a best example where we can see the trisomics. And Gish, whenever you have the polyploids in the form of the more number of sets of the chromosomes A, B, and D like structures, and we can give the different genomes with the different colors. So like this, so this is one of the karyotypes of the human beings. So here is the, I would like to give the emphasis of the evolution of the wheat crop. So wheat, it is one of the most staple food in most of the regions of the world. The wheat is basic chromosome number we can see in the form of the 7 chromosomes. So in the form of the Triticum monococcum, which is having the AA genome, and it is having the 14 chromosomes. So it is accidentally crossed. It is not a man-made polyploids, allopolyploids. So it is the in the form of the uh, 7 chromosomes plus B genome is coming into cross in between in the form of the AB genome. It is in the form of the a diploid like structure, but it won't form the seed. By converting in the form of the ampidiploids in the form of the Triticum turgidum, we can see the chromosomes in the form of the tetraploids, that is in the form of the 28 chromosomes. So generally in the crop plants, when you cross any two different species, we won't expect some extent seed formation. But in case of the Agelopsa speltoides, one of the B genome, fifth B genome, which contributes homologous pairing rather than the homeologous pairing. So because of that, we can see the one of the tetraploid plant in the form of the Triticum dicocoides or Triticum turgidum. So fifth B genome, which contributes in this wheat crop, which is useful to pair only in the form of the seed formation homologous pairing rather than the homeologous pairing. What is the difference between homologous pairing and the homeologous? A is paired with A and B is paired with B, that is known as the homologous. A is paired with B and B is paired with A, that is considered as a homologous pair, homologous pairing. So that will be contributed in the form of the fifth B genome, which pairs only homologous regions. So another species of the grass species, Agelopsa sparosa, which is a drought tolerance, which we can see wheat having the D genome, and in the form of the F1, that is the ABD genome, and it is only one set of the n number of the chromosomes, and it will be diploidized, and we can see the 6x in the form of the 42 chromosomes. So wheat is one of the best example. It is all of polyploids, and it is not a man-made crop. So we can identify the species level in the form of the diploids, tetraploids, and the hexaploids, and the chromosomes morphology some extent we can see. And here is also we can see the in the form of the wild grass and the in the form of the cultivated, and they form in the form of the tetraploid, that is the Triticum turgidum, and it is crossed with another wild type, and it is in the form of the hexaploid in the form of the wheat crop, which is evolved. It is not the man-made crop, it is evolved on its own, and it's based on the phenotypes as well as we can identify based on the chromosomes. So like this, it is one of the best example what I observed in the wheat. We can also have the some more maps in this figure. Further, we can see the spike nature of the hexaploid, maybe having the very big sized seed and having the in the form of the in the form of the hair-like structures, which is maybe lengthier than the normal cropland. So like this, wheat is one of the best example where we can see the hexaploid cropland in the form of the six hexaploid 6 into 7, that is in the form of the 42 chromosomes in the form of the ABD genome. And by using the GISH genomic in situ hybridization, we can identify each chromosome based on the genomic level, and we can determine this crop. So in addition to that, another crop plant which is evolved in the form of the Brassica members. So in the Brassica, we can have the three basic species in the form of the Brassica nigra and Brassica raffa and Brassica oleracea. So you know the Brassica oleracea in the form of the cabbage, and Brassica nigra, it is the black mustard. So it is having the BB genome n equal to 8. This is the 8 chromosome, one of these species. And another species in the form of the Brassica raffa, which is having the n equal to 10 chromosomes in the form of the B genome and A genome. And the Brassica genetica, one of the species, which is the brown mustard, it is the Indian Brassica variety. And you can see the chromosomes maybe here. Generally, when you cross two plants, maybe equal number of the chromosomes, 8 plus 8. But here we can see the 8 plus 10. So in the form of the 18 chromosome, it is a new species origin in the form of the Brassica genetica. So we can see the increasing order of the chromosome number in this Brassica varieties, 8, 9, and 10. So like this, we can see the in between a cross in Brassica nigra and Brassica oleracea, we can see the in the form of the Brassica ornata with BBCC genome. And another species is origin amalgamation of these two genomes, CC and A, in the form of the Brassica napus, in the form of the uh, rapeseed. So this is in the form of the U turn. We can see the from the three crop species by amalgamation of the genomes. So we can see the chromosome variation number, and we can see the in the form of 8, 9, 10. So increasing order, and also we can see the in the form of the in the form of 17 and 18 and 19 like the species. So some extent the chromosome number is increasing in the evolution of the different species within the Brassica genera, Brassica genus. So here we can see the Brassica nigra and the Brassica genetica. So nigra is the black mustard, and Brassica genetica is the brown mustard. That is the Indian mustard. The Indian mustard is a tetraploid, not the diploid. So it is the union of Brassica nigra with the Brassica campestris in the form of the AB genome. And you can see the Brassica genetica, which is contributing much in the oilseed in India. So like this Brassica oleracea, it is a diploid, and it is in the form of the 9 chromosomes, and we can see that is the cabbage and rapeseed. Like this, we can see the different members. So this a good example where the genus, which different species, some extent in the form of the diploids and the tetraploids. So by amalgamation of the different genomes, arise 3 different new species. That is very interesting what I observed. So you can see the Brassica nigra distribution and genetica some extent, which is the Indian continent, and Corneta we can see the in the or maybe in the old world. Like this we can see the different regions, and we can see might be they are adopting, and we can see the different regions of the world. So this is also, so here I would like to explain one of the cotton species. In this cotton, we can see the in the form of the Gossypium species, there are 2 genomes in the form of the A genome and the D genome. So A genome may be old world in the Gossypium Herbaceum and Gossypium arboreum, and which is there in the form of the originated hirsutum by union of the one of the New World cotton. So we can see the A genome, which is the Old World cotton. D genome, this is the New World cotton. Here we can see these two species. Just now we learned that the chromosome number may be same here, but we can identify the species based on the chromosome length in the form of the Old World and the New World. So here the Gossypium herbaceum and in the form of the Gossypium other barbadans, which is a cross in the Old World cotton and the New World cotton in the form of the American in the form of the Gossypium raimondi. This is the American variety, also known as the in the form of the New World cotton, Gossypium herbaceum. That is the Old World cotton. So you can see the chromosome number is same here. You can identify the 13 chromosomes in each species, raimondi and the uh, herbaceum. But you can see the here somewhat it is written in instead of short long chromosomes here, it is written in a negative in a different way, small for it is here long chromosome, Old World cotton, which is containing the long chromosome, and the New World cotton, that is the American cotton, that is the short chromosomes. So instead of say small and large, we can say short and long chromosomes. Here, the Old World cotton, which is having the long chromosomes, and the New World cotton, which is having the long chromosomes. So in the form of the 2n plus 13 plus 13. So in the F1, we won't get in the form of the, it is in the form of the univalents. So somewhat you are talking about the fertility and pairing of the homologous chromosomes. So here two different species, and there is no homology in the chromosomes because you know the short chromosomes and the long chromosomes. Until you have the 26 chromosomes, you are thinking that these are the two different species morphologically some extent, but cytologically you can see the chromosome size, not the number. Number is same in the two different species. Based on that, the new cotton that is originated in the form of the tetraploid Gossypium hirsutum. It is actually New World cotton, and it is known as the upland cotton, which contributes the 85% of the cotton in the world. The worldwide cultivated cotton that is in the form of the Gossypium hirsutum, which is having the 13 long chromosomes, 13 short chromosomes. Another beauty here, either of these two species, the fiber length is less. But in case of the Gossypium hirsutum, it is longer than either of these species. So you know that the Gossypium species, which produces the cotton, and we can use as a cloth. In addition to food, we have the three things we need in the form of the food, shelter, and cloth. So Gossypium species which contribute the cloth. So most of the persons love with the cotton cloth rather than the synthetic cotton. So most of you wear the cotton shirts and cotton saris, but not the synthetic. So here hirsutum, which is having the trichomes. So most of the botanists may know or they have limited knowledge. So the trichomes generally contributes less important, but taxonomy, which is having the more important, here the commerce also says that the trichomes of the cotton is very important. The seed hairs are the trichomes. It is in the form of the unicelled trichomes and containing the chemically cellulose. So this is one of the best example to see the anatomical and we can do some chemical studies in the cellulose. That is the best example to study the cellulose in the cotton. So here we can see the this is the D genome, it is the short chromosomes. This is the New World cotton, D genome. You can see the 13 chromosomes. They are shorter than the A genome. This is the long chromosomes. So instead of smaller and uh, larger, we can say short chromosomes and the long chromosomes. So the terminology, what I am feeling, it is we need to use short chromosome, the size is less. Long chromosome, the size is more. So here these two crop species, that is the old world cotton. So that is one of the Gossypium species. The Indian cotton, that is the Gossypium herbaceum or Gossypium arboreum. The American cotton, Gossypium, maybe Raimundi and other species. By union of these two species, it is produced in the form of the new species, that is in the form of the Gossypium hirsutum, that is the upland cotton. And you can see there is no hair, there is also short hair. So what is the coming hair in the seed? You can see the long side hairs. So it is one of the QTLs, quantitative trait loci in the crop plants, which contributes, instead of 1 plus 1, you have 1 plus 1, 2 plus 3 like that. You can have the 3 genes and it is longer. So sometimes you can see some of the parents, both father and mother, they are not longer, they may be short. But the child coming in the form of the siblings, they are taller than the either of these mother, father. In the same way, you can see the cotton fiber, which is coming from the seed heads, longer than the either of these species. In the same way, you can see the human beings. Sometimes both the father and mother is shorter, but siblings which are taller than the age of the parents. That indicates the QTLs, quantitative trait loci, which enhances the uh, uh, character of the specific phenotype or yield like that we can see in the crop plants. So this is the best example where the chromosome number is same, and we can see the 13 chromosomes, we can see the different species, but we can identify the origin of this crop plant by the amalgamation of these two species, and it forms the new species, that is the AD genome, and the basic chromosome number 13 into 4, 52 chromosomes we can see. So like this, we can see this is the wild cotton, and which is the cultivated cotton, which is having the longer fiber. So like this is the wild, and this is the domesticated cotton. So most of the world cultivating cotton, and we nowadays modified at rather than genome level, and we have in the form of the transgenic plants. This is also semitotic studies. So in addition to that, I would like to explain briefly about APG classification. It is generally the plants are classified based on the morphology, anatomy, and embryology, and phytochemistry, and more strongly on molecular studies with reference to the DNA sequences. So this is one of the APG classification. Now it is very familiar. It is taking the DNA sequences and from the chloroplast genes and one gene coding for the ribosomes from the nuclear genes. So here they are taking into consideration in the cytoplasmic genome. And also one plant is coming from the other plant, that is the concept from the existing plant, new plant is forming. So the cytoplasmic genome, which contributes in the plants by the mitochondria as well as the chloroplast. But generally in the cytoplasmic genome, which is considered for the classification angiosperm phylogeny group, they took the chloroplast genome because the chloroplast genome is highly stable, and at the same time the length of the genome is short or less in length. Maybe that is 150 kb or 200 kb, in between 150 to 200 kb. So the chloroplast genome, and one gene is taken from the nuclear genome. that is from the ribosomal DNA. So that is taking the 3 genes all together in the form of the chloroplast, that is taking in the form of the ATP-B gene, that is having the 1450 base pairs, and another gene, ribulose bisphosphate carboxylase enzyme. So we can have the uh, RBCL maybe having the in the form of the 2 types, large peptide and small peptide. They took the large peptide in the form of the 1428 base pairs. So like this in the form of the ATP, in the 1450 base pairs. In addition to this, these 2 genes from the chloroplast genome, in addition to nuclear genome, that is in the form of the, you know, that ribosomes, which contains the different RNAs. So 18S rRNA, that is in the form of the DNA sequence by taking these things, which is doing the classification. So molecular biology, which in addition to the cytology, we can also concentrating nowadays APG classification in the form of the, earlier we thought that monocots are evolved, but monocots which are placed in between uh, primitive dicots and the evolved dicots in the form of the monocots which are there in between. So like this we can see, in addition to morphology, phytochemistry, anatomy, we can going from the cytology to now in the molecular basis. So like this we can see the different things. All together we studied the what is karyotype, what are the different karyotypes are there, and how to distinguish the different karyotypes, like in the form of the symmetric and the asymmetric by taking the semitotic studies. And we can distinguish the species level chromosome number, or in the form of the subspecies level, or in the form of the, we can differentiate the genus levels and family levels. And also we can see the, sometimes I observed very interesting, I observed in case of the cotton, where we can see the in the form of the long chromosomes and the short chromosomes. So the old world cotton, which is having the short chromosomes, and old world cotton, which is having the long chromosomes, and the new world cotton, which is having the short chromosomes, till the both the species which is having the same chromosome number in the form of the 13 chromosomes. So like this, we can distinguish the species level chromosome number. Sometimes may be same, but size of the chromosome is differs. Best example where we observed in case of the cotton. And we observed in case of the Brassica members, the increasing order of the chromosomes 8, 9, 10, and also 18, 19, and 17 like structures. We observed different species. So with this, I will conclude this session. I am thankful to Professor Vijay Bhaskar Reddy and other organizers in this session.
Yeah. Thank you so much, sir. I think there are some participants raise their hands. Sadhana, please look into that.
Okay.
What is the role of other 32 chromosomes of monotropics hypotopics?
Sir, can you turn on your video?
What is the?
So this is a question we got from the participants. What is the role of other 32 chromosomes of monotropics hypotopics?
It is one is the hexaploid, the other one is the diploid. Based on that, these two species are segregated within the same species.
That's it, sir.
Dr. Jaladhar?
Dr. Jalandhar, sir, if you have any question, please unmute your mic and please do ask.
Dr. Jalendra, do you want to interact with the
Participants can raise their hands to ask the question. Dr. Jalendra has raised his hand. Sir, kindly unmute your mic and pose the question.
Okay. There are no questions. Then I will propose a vote of thanks to Dr. S. Karnak Reddy. Dr. S. Karnak Reddy recently retired from his service, and as framing the structure of this course, plant taxonomy course, already I told you we felt like lacuna in one of the important aspects, that's a cytotaxonomy. And immediately we approached Dr. S. Karnak Reddy sir, and he readily accepted. And though his research background is different, but he accepted and he presented well. Each and every slide is very informative. And of course, what he presented, that various things about the chromosomes, their number, and their morphology, definitely they play a very important role, great role in the identification of the varieties, because so many varieties are producing day by day by the different institutions, and to identify those all varieties, the cytotaxonomy plays a very key role. Really, sir, you have given a gist of cytotaxonomy, and you enriched our participants' knowledge by sharing the important taxonomic aspect. That's a cytotaxonomy. Thank you once again, sir.
Thank you,
also,
okay.
Yes, sir. Also, thank Sadhana and Srijan coordinating the sessions, and participants. There are nearly 250 participants every day. They're coming and participating in each and every session. They're listening very patiently. We thank them from organizing committee, and time to time we are giving evaluation forms. You're also we are getting a good cooperation from your side also, because running a course is not a simple thing. For this, we are taking much pains. The support of the Dr. Shashikala Madam and Dr. Ramanchi Nageshwar Rao. We have we framed this structure. Today, this everything is materialized and continuing with all the sessions. So, participants, we request you all continue same tempo and cooperate with us. And your cooperation is much required. And Dr. Ramanchi Nageshwar Rao, to share something with the participants, please.
Sir, sir is not on the screen, it seems.
Madam?
Sir is not there, it seems, sir.
He is there.
Okay.
Yeah. Good afternoon, everyone. Almost we have completed 2 days, yesterday and today. Because of the technical glitch, we could not start the sessions in time. It is not from our side or your side. Because of technical problem, we are starting the sessions little late. Otherwise, we would have done in a systematic manner. But from our side, we will try to improve also. From participant side, we have got a very good response. Your response is encouraging us to continue the program very actively for the next remaining days, almost 4 days. There are queries, requests from the participants asking that time and again you're asking for the feedback form. So we are also working simultaneously. We are listening to the classes. We are arranging the resource persons, and also we are working for the generation of the Google Forms. As you know, as you are very much aware of that, we are making the questions, setting the questions. So don't be anxious about the questions because they will be very simple questions. But because we are running a course, it is mandatory from the university side and also UGC side. There should be an evaluation method. We have got responses. We have got the answer sheets also. And there and then you will be knowing how many marks you got. But we are not considering any marks for the giving the certificates. We'll give the certificate for the all participants, those who have attended course completely without missing any session. That is the only criteria, not the marks. But very carefully listen the class and try to answer all the questions. And we are also have in the schedule, we have a one day question paper will be there. About 20 questions will be there that each and everyone has to write that 20 question paper, and we will give the ample time. We have a request from the a few participants that you need much time, but we are giving very few questions. So try to answer within a couple of hours time, or else you take 1 hour extra. But we will not repeat the same question paper next day. And that is what we decided, and try to cooperate with us also. With these small announcements, if anything is there, please make a chart in the box. And we request you don't simply chart good morning, good afternoon, good evening, excellent presentation, something. You pose the specific questions so that it will be useful for the organizers to improve, even for the resource persons to interact with you. Okay, so we are welcome the questions in that idea. Okay, thank you very much. Thank you very much, Vijay Bhaskar Reddy sir, Shashikala madam, all the technical team. Thank you very much.
Sadhana, you are telling one of the students is asking about the trisomics.
Yes,
Hello?
yes, sir. Yes, sir.
That is equal to n number of the chromosomes. If it is a diploid, that is n equal to 7, for example. 7 trisomics are possible. So in case of the Datura stramonium, there are 12 pairs of the chromosomes are there, and 12 trisomics are possible. In humans, there are 23 pairs of chromosomes are there, 23 trisomics are possible. So if it is hexaploid, you can expect like that the number of equal to, if it is wheat, for example, 7 basic chromosomes, 7 into 6, 42. 42 trisomics will be possible. So the trisomics, irrespective of the plant, is equal to n number of the chromosomes. So haploid set of the chromosomes. If it is hexaploid, you can take the n into 6.
So, thank you so much, sir.
Thank you, sir. And Maunika, would you like to share something? You raise your hand. Please unmute yourself and talk.
Unmute here.
Monica, please unmute your mic and pose your question.
Sir, some participants are not aware of raising hands, sir.
Yeah, okay.
So if they pose a question in the chat box and then they raise their hand, we'll take the question, sir.
Okay, that's better. Yeah. Tomorrow we'll have 3 sessions. 3 speakers will be with us. One is Dr. Narsimhan. He will deal the classification systems, including APG system. And also afternoon we'll have again Dr. Nandikar. He will go with the web resources for taxonomic research. And also from Botanical Survey of India, Dr. Swami will be there, and he will deal all the tools, taxonomic tools like GPS, herbarium, and etc., which are used for the improvement of taxonomy or deal with the taxonomy. So all sessions will be start by the time. Please be on time. Tomorrow morning session will be start by 10 o'clock, and link will be available from 9:45 onwards. Afternoon as well, 2 o'clock. Link will be available from 1:45 onwards. Please be with us, and see you tomorrow. Thank you, one and all.
Sir, before we conclude, I have 2 words to say, sir.
Yeah, over to Sujan.
Dear participants, you kindly do not download anything that is shared in your chat box link any links other than from the panel, because we have identified some technical glitches because of hackers. I have identified them. We have reported to the Zoom also. So the morning session was also because of the same thing. So even in YouTube also we have found some glitches because of the hackers itself. So I request you all, please be secure on your side. Do not share any link or do not click any unnecessary links whichever comes in the chat box, because sometimes that may not be seen from the panel side. So we have ensured the safety to the most, but still this is a kind of advice to you all. Make sure that you only click the link shared from the panel, not from anybody else. So we are not going to share any documents like PDF or any other PPT files in the chat boxes. We will only share two kinds of links. That is one YouTube link, other one is a Google form. Other than these links, if somebody is sharing any other link, do not click it. That is from my side. So if it is possible, you just come on YouTube, and not on YouTube, just on Zoom itself every day. So we have identified some abusive images in our YouTube today. So we have covered that also. But if something happens tomorrow on the same Zoom platform, kindly just leave the meeting and come back again and report to us at the earliest possible, because it may be embarrassing for people also, but this is a problem with online programs. But ensure that you reach the message to us so that we will put them down. Thank you all.
Madam, Sasikala Madam, Jeptara, would you like to tell anything?
Sir, dear participants, please cope up with the technical issues, whatever the instructions which are coming from the organizers as well as the technical team. We need not have any regrets, or we need not lose our time, precious time, because all the speakers are eminent speakers. Their time is very valuable. So that's why what we request from our organizers are, please cooperate with us. Sometimes some technical issues are coming with different connectivities and all, and also power problems. So that's why please cope up with us. This is one thing. And another, coming to the Kannaka Reddy's class, it was very, very elaborative and very informative and very useful, especially in the area of cytogenetics. We had very good class. Majority of the participants, they wrote very good comments on your talk, sir. Thank you very, very much for you being here, sir, to enlighten our participants. And also, sir.
Okay, ma'am.
Thank you.
Yeah. One more thing, to all the participants, the panelist, the panel, we have Sadhana, we have Srujan, Karthik, and these are all technically they are supporting us, technical crew, but they are all research scholars and students of Osmania University, environmental science and different disciplines. So we are very much thankful to their services. To know the all the participants, I am telling this. We are very much thankful to all the technical team. And one question I want to answer from the chat box that is not subject related. One of the participants said it is almost 5 hours or 6 hours are going on in the on screen. So it is very difficult to manage something like that, he was telling. But from our side, it is humble request. We need to complete the required number of hours or classes to fulfill the criteria. That is why we have arranged 2 hours in the morning, 2 hours in the afternoon. Wherever possible, we have arranged 1 more hour, 3 hours. So we are trying to accommodate all the lectures, lessons, which is necessary to qualify as a short-term course by the UGC or any recognized organization. That is why we have accommodated 5 hours somewhere, sometimes 4 hours sometimes. So keep in that mind, please make your alternative arrangements your mobile data is not enough, please keep your mobile data as well as Wi-Fi connectivity to your laptop or computer, whatever it is, iPad. So make sure that the program will go in a smooth manner. So we cannot shortcut the complete program as of now. Thank you.
Please introduce Karthik also.
Yeah, no, Karthik also a research scholar, Osmania University. He is also student of the Dr. Shashikala Madam. Is also one of the backbone of the entire 1 week program.
Sir, I would like to talk about Karnaka Reddy sir's session.
Thank you, Karnaka Reddy sir, from my side also. Yeah.
Sir,
Sadhana, please.
Yes, sir. Sir, Karnaka Reddy sir, I was so much overwhelmed by seeing the thank you slide of Karnaka Reddy sir's session. The thank you slide itself was an overview about the entire session he gave. He literally connected each point of the session to the thank you slide. He's the first person who has explained the thank you slide for 1 minute, sir. That was really complimentable. So thank you so much, Karnaka Reddy sir.
Yeah, thank you all. Srijan.
Sir, I'm ending the meeting, sir. Thank you, participants.
Yeah,
Kindly come back on time tomorrow. See you all. Bye.